Right. So thanks for good questions in the break that helped clarify a few things. So one is, one of you emphasized that, yes, so far we've not been using coordinates at all. This is correct. So here I try to plot the same nodes, 1, 2, 3. Let's say this is 1, 2, 3. And then the fourth node, well, I've laid it out in the other direction, but I could also, this node number 4, I could also lay it out like so. Yeah. So no coordinates were used. In a social network, there are no coordinates. In a molecule, of course, there are. But the way we've discussed it so far, these coordinates are not used at all. It's only this chemical connectivity which is being used. And that will change in January when we discuss these architectures that really know what to do with coordinates. Then for me, the 1 is not the identity matrix, but it's the vector filled with 1s. And here I forgot the transpose. And here I just wrote C of A, but this cannot be any function of the adjacency matrix. It must be one which is permutation invariant. And then I also, for consistency, I added the adjacency matrix there and there for a bit more generality. Anyway, these are not the most general expressions. You know, this is e.g., because in these examples, I always assume that you have node features, but you might also have, in addition, edge features. And those are sometimes used to great effect, like in message passing neural networks. Those I had not written here. Okay. So in part 2, I wanted to talk about random stuff that I find interesting. And one of these is molecular biology, because I don't cease to be amazed by the sheer complexity of what happens in a living being. So here we look at a tiny part of a single cell. So we are made up of lots of cells, but the fact is that even a single cell we don't understand yet. And the reason you see, or part of the reason you see depicted here, this image, by the way, it's a very nice scientific presentation, in the sense that this was not drawn, but these are really proper coordinates that were used. Yeah? So the artists here, they use proper coordinates of peptides or of proteins as extracted from databases, and it's only the arrangement of these which is artistic. And here, you know, where there's void, in real life, there is not void. Yeah, it's packed with stuff, and it's just lots of stuff here that has been omitted to give you some perception of depth. And well, what we see here is, you know, some transmembrane channeling. We see a bit of cytoskeletal structures. This must be part of a mitochondrion. And a biologist would be able to, you know, talk about this picture for an hour, which I cannot. You know, all these transmembrane proteins that we have up there. So it's an amazingly complex machinery. And one particular structure this brings forth is, of course, the human brain. And you know, let's look at that a little bit. So the brain, you know, it's around a kilogram. Homo sapiens, I mean, the Neanderthals, by the way, they had a bigger brain than us. And there's a debate about, you know, is it like in whales that also have bigger brains but are not thought to be more intelligent? Or is it maybe the fact that they were more intelligent but just less apt as less apt at social interactions compared to Homo sapiens? And well, if you are living, you know, as a sort of, back in those days, humans did not have much of a technological edge relative to predators, and so the social group was really important. Yeah? And there is this is speculation, but there's some serious speculation that what gave the humans an edge over the Neanderthals was the fact that they could maintain social relations with a bigger group of people. But so this is around, you know, a kilogram, and if we don't understand a single cell, then of course we don't understand this organ as a whole. We can look at it at a macroscopic scale. So this is what anatomists have been doing for a while. Or here you can do something called tractography using diffusion tensor imaging, which is non-invasive. So you could put one of us into an MRI and create that kind of picture, and we would walk away alive. But unfortunately, much of the brain to us is only accessible using destructive techniques. So here we see what the dendritic arbor of a single neuron looks like. This is one of the famous images from Cajal, who couldn't quite decide if he wanted to be an artist or a scientist, and then became both in a way. And well, I am, so we have in the human brain around 10 to the 11 of those, and each one takes 10 to the 4 inputs from synapses from other neurons. So that's, and then we have a single axon projecting out. So that's a pretty phenomenal machine, and also computing machine. Yeah, so it's a pretty serious supercomputer which we have between our ears, and it runs at only 20 watts or so. Yeah, so this is very far from what technology has accomplished. Well, and if you zoom in further, here we see adjacent slides in depth of a single synaptic contact. So here you see this dark thing that would be some, well, the proteins involved on the postsynaptic side in particular, which are dark. And here you have these vesicles containing neurotransmitter, which in chemical synapses can, you know, merge with the membrane and release neurotransmitter. And by the way, this kind of architecture that we use in our part of the evolutionary tree, that's not what it looks like in insects. Yeah? So insects have found a maybe more flexible and maybe more efficient means of processing information at the cellular level. So the way it works with us is this neuron collects input from these dendrites, and if there's enough input, this triggers, you know, pulse, which then travels along the axon and projects to other neurons. So you could argue this is, you know, sort of fairly hierarchical processing. And insects, they don't have this separation of here we gather input and there we send output. But in insects, these are both sending and receiving. So it means you can have interactions at a tiny part of a neuron without needing to first send it to mother central, where, you know, a decision is made whether or not to trigger a spike. And it is argued that this is responsible for a couple of things. So for allowing insects to do so much compute in so little space. Yeah, because if you think about the fruit fly that sits on your banana, you know, the fruit fly in itself is pretty small. Its head is much smaller still. The biggest part of the head are just the eyes, those compound eyes, and between those compound eyes sits the brain. So it's a really minuscule structure. And this really minuscule structure, with only 130,000 or so neurons, it allows the fruit fly to see, to smell, to fly, to mate, to evade danger, to find the banana, and you know, lay eggs, all the things it does in its life. So, and that happens, you know, the fruit fly as a whole, I was arguing, is pretty small. So if you think of how much space there is and how much energy can it afford to use for its computation, it's remarkable what it achieves. Yeah? And this is thanks to its very flexible, at the microscopic level, neural structure. And so evolution has found a couple of ways to process information. And okay, both here uses neurons, but they're still quite dissimilar. It's just how evolution has found different ways of also realizing our other senses. Yeah, so some of the best eyes that we have in the animal kingdom are the eyes of these deep sea octopuses, and you know, they have these huge eyes also because there's very little light down there, and it was clearly reinvented by evolution, because if you look at how the eye in, for example, a mammal develops, then the eye is a protrusion of the brain. Yeah? So you have, during the embryonic development, you have this stalk, ein Stiel, yeah? You have this stalk which starts projecting out from the brain, and then eventually reaches, you know, the front side of the face, and there induces the formation of an eye and so on. And in these octopuses, it's the other way around. There's, you know, some pigmental change happening on the skin, and then it then starts building neurons that find its way towards the brain. Yeah, so very, very different mechanism of realizing an eye, and both work well. And speaking about the eye, I mean, we're pretty well equipped if you look at the animal kingdom. Yeah, so we have, if you look at the fovea, our area where the receptors are most tightly packed, we have around, per square area, around half as much as an eagle. So the eagle is around the best that we know. They have twice as much, but we have 7 times more than a cat, for example. And that I didn't realize, because cats have beautiful eyes also. So we see a lot better than cats. And by the way, the Neanderthals saw better than us humans, because they were, they had bigger, it's thought that they had a bigger visual cortex that helped them process low light information better. And it's argued that this was helpful in the Arctic areas where they lived very long winters. So it's good that not all that was lost on us, because all of us have between 0 and 5% Neanderthal. 0% if you're mostly African, and 5% or so if your grandparents lived more in the north. All right, but we were talking about the eyes, and we were talking about the brain, and about synapses, and so there are many ways to try and make sense of what happens in the brain, but one of these is in terms of connectomics, And I wanted to highlight a little bit the role that Winfried Denk played in this because he did that piece of work in Heidelberg. So Winfried Denk is the developer of the two-photon microscope. He always liked building stuff, and he previously worked in MRI, but in MRI it was difficult to build, your own. And so he eventually turned to microscopes, where it was easier to fiddle around and take it apart and reassemble it. And so his first major accomplishment was the two-photon microscope, and his second major accomplishment in microscopy was a serial block phase scanning electron microscope. So this was a device where you would take a sample, you would turn it, basically you would stain it, you would turn it into plastic more or less, you would make it very, very flat, and then take an electron microscopic image of the surface and then shave off the top layer, even with a diamond knife or with a focused ion beam, and then take the next layer and then take the next layer and so on. And this he did in Heidelberg in the early 2000s. And the aim was to try and understand how the brain is wired up. And now I see this, but not you. I need to bring it to the other screen. All right. And so he pioneered this kind of staining using horseradish peroxidase. So what we see here is a little bit of neuropil, and stained are only the membranes. And this is now an artistic rendering of the kind of data that he would give us in the early 2000s. And you see here on a scale of 1 pixel is around 5 nanometers or so what this looked like. All right, and now came we turned this raw data into, that's a regression result. Here we try and decide for each pixel, is it membrane or not? And this movie is from 2008, so this is when we did that. That was using shallow classifiers, random forests at the time. And this was possible for volumes of the size that you see here. That was not a computational limitation, that was what could be imaged in high quality in one go using this kind of microscopy. And then we would build superpixels, and then we would piece together the superpixels that belong together. And then we would trace out neurons. Here it looks like it's, this is not a neuron, it's a tiny part of a projection. Here it looks like this would be done one at a time, but of course it really happened all at the same time. So this was parallel processing. And so you could look at different subsets and at more different subsets. And when we bring back all neurons, you again get this impression of how magnificent the complexity is in the tiniest part of a brain. So this was restricted to fairly small parts of the brain. And I want to introduce to you a gentleman who did this on a much bigger scale. And I want to do it both because he produced outstanding images and also because he did that single-handedly as a PhD student. So sorry, I should have put this into mirroring mode. All right, so here we see Davy Bock. And when he was a PhD student, he custom built an electron microscope. This was a few years before that picture was taken. This custom electron microscope was like 4 meters in height. On top was the electron microscope. It sat on huge pillars. And at the bottom was this whole device for putting in one sample after the other, because he also used this shaving technology. And this was, an outstanding instrument that he built himself with his own hands in a PhD, which took 5 years or so. And he later became group leader at the Geneva Research Campus. And that's when that picture was taken, and that's when he built the same device, only prettier and so on. And he's sitting now on the top floor. So there's a hole in the ground, and what you see is the top of the instrument, and it goes down another 2 meters or so. And now this is the picture that you saw from Cajal, and this is drawn at the same scale, the kind of picture that Davy would acquire. And I want to show you a little bit what this instrument has been capable of. So I'm zooming in. What we see here are well, this is a cell body. And we zoom in, and we start seeing mitochondria, like here or there. Mitochondria are these organelles that have membrane stacks, and they're often called the powerhouse of the cell. And these mitochondria, I would bet that the mitochondria that you've seen in your textbooks when you went to school, and even in the textbooks that are published nowadays, these mitochondria, they are like, sort of thick sausages. Like you have a sausage this long, and then that's a picture of the mitochondria, and it is cut open to show the membrane stacks. All right. Okay, I see a lot of nodding. This is not what mitochondria look like. And that was discovered thanks to this kind of imagery. Mitochondria look more like what I described to you, the minimum of a neural network. I claimed, it's not a point in parameter space, it's this manifold, it's this network. And this is what mitochondria are. So not so much a network, but they're very elongated structures. So you should think of mitochondria not as sausages, but as spaghetti extra long that weave their way along the cell. And this you don't typically see in a single image. Yeah, so I mean, electron microscopy was a great invention. And then looking at pictures like that was fantastic. And then people, well, from that, they started building an intuition of what does a mitochondrion look like. But here in this conventional type of electron microscopy, you did not have the serial sectioning that Davy's instruments would allow. You lacked the third dimension, and people were lacking the intuition from those cross-cut images to imagine what it might look like properly in 3D. And well, another word about mitochondria, they're interesting because the most plausible hypothesis is that these are, so we are eukaryotes, but probably what we see here is an example of endosymbiosis. So this is a different life form. I don't want to call it alien, but a different life form. And this different life form, a few 100 million years ago, decided to coexist with our cells. And this is why now, in each of our cells, we have mitochondria who are a really important part of our energy household. they, in this what is presumed to be an endosymbiosis, they have not decided, to go in all the way. They've decided to still pass on their own hereditary information. Yeah, so the information on how to make a mitochondrion, it's not stored in our DNA, or let's say in the DNA, which is in the nucleus, but the mitochondria have their own. They have their own recipe of how to reproduce themselves. And this is one of the things, one of the sources of information besides DNA that is passed in a zygote, so in the cell that will eventually make all cells. And just curious here, our small pub quiz question. Let's look at the amount of DNA that we have. So let's say a sperm meets an egg. In the sperm, there's one. So in our normal cells, we have 2 copies of each chromosome. In the sperm and in the egg, there's just 1 copy of each chromosome. What do you think? So you know that this is DNA. You know its base pairs. We can trivially translate this into how many bits this translates to. So what do you think, how many bits of information or how many bytes are in a haploid, in a sperm cell or an egg cell? Is it a gigabyte? Is it a terabyte? Is it a petabyte? Who thinks it's I will just ask for hands up. Who thinks we have a gigabyte of DNA to code a complex being like ourselves? Who thinks it's a gigabyte? All right. Who thinks it's a terabyte? Who thinks it's a petabyte? Okay. So DNA alone accounts for around 1 gigabyte. And 1 gigabyte, I mean, I don't even have a USB stick that small, right? And imagine how complex we are. Yeah. So this is outstanding how nature has managed to somehow compress information, or to how to bootstrap complexity out of very little. And on the other hand, to be fair, DNA is not everything that we pass on when a new individual is created. There is the DNA, but there is also different methylation states and so on of DNA. And this is has long been, or it took a long while to discover that in the first place. So for a long time we thought the DNA is all there is to know about passing of information. But this extra information, it's difficult to estimate it exactly, but it's thought to increase the amount of information that is encoded by maybe tenfold. Yeah, so we're already talking 10 gigabyte and not 1. And then comes all right, then there's a little bit of information in mitochondria and so on, but that's not so much. But there's the 3D structure itself. And now in the cell, or let's say in the dividing cell, we don't need to know the position of each and every atom. Yeah, so it's not like number of atoms in a cell times 3. That's not the amount of information we need. But somewhere between where individual atoms are and ignoring spatial information altogether, this is where the truth lies. And there is apparently a lot of information stored in precisely, how this machinery in the cell is arranged. And so it's difficult to estimate for how much information that it counts more precisely. But very, very vaguely, super vaguely, and this could be off by orders of magnitude, it's maybe a terabyte of information that we pass on. And even that, feels very little if you think about 1 terabyte compared to the complexity of our 10 to the 11 neurons and so on. Yeah, and each neuron has 10 to the 4 dendritic contact, so that makes 10 to the 15. And this is just the brain, and there's the rest of the body also. So apparently nature has invented amazing compression mechanisms, and nature has invented also, good ways to reuse building patterns, like our brain, We said it's around 1 kilogram, but you can take the cortex and you can, it has these folds that's to increase surface area, so to fit a big brain into a small head. But if you unwrap it, all these wraps, it's around the size of this table, the cortex, and it is made out of cortical columns. So you can think of one cortical column being one processor. So we have this massive parallel processor. And so the ambition in understanding the human brain is to understand a single one of these cortical columns. So to understand one processor at first, and then later on how they will act together. And by the way, you know how difficult it is for you to imagine the smell of a lemon. I'm not saying taste. When I say taste, it's already, maybe you get a little bit of water in your mouth, but the smell is really hard to imagine, or the smell of a rose or anything like that. And the reason is that smelling and tasting, in the evolutionary sense, they're old senses. So they are relatively far from our advanced processing in the neocortex. And this is why it's so difficult for us to name smells and tastes. So you might have done this sometime in school. You get a yogurt and the color has been mixed into there, which is not supposed to be there. Whatever, you get a blue liquid, and then you're supposed to tell that this was strawberry. And this is because, you're unable to name it because, the color doesn't match what you sense, and what you sense is too remote from your higher processing areas, which is why you cannot name it. You recognize, there's something in this dish, but you cannot name it because evolutionarily there's so much time in between. But we started this journey here, and we were zooming in just to see what this kind of microscopy allows. And we talked about mitochondria. We see more synapses here. There's presynaptic vesicles. There's the membrane. And I like to talk about these because Anna Kreszuk, who was a PhD student in the lab and is now at the MBL, I mentioned her before as a group leader. She was the one who developed the first automated synapse detector on this kind of imagery. And we can zoom in further, and now we can go in depth. So now I'm not zooming in, I'm moving in depth. And you see that these images here are relatively, they're not as smooth as the data that you saw from Winfried Denk, which is why this kind of data was much more challenging to segment, and which is why the community, this data here was acquired, I think, in 2018. Most of the community gave up on this data set as it being too hard to segment. And but some people didn't give up. And by the way, here now this is around 1 millimeter, and the size of a mouse brain is several millimeters, so cubed. So doing the whole brain at this thing for the smallest mammalian brain that we can find, that's a major endeavor. All right, but some people didn't give up, and one of these people who did not give up was Sven Dorkenwald here. I am highlighting him here because he's the first author, and because he was also sitting on one of these benches. He was a physics bachelor student in Heidelberg, and then did his master's in computer engineering, and then moved on to work on these kind of tracing problems. And this so-called Flywire Consortium, and you see this was very recently published, along with 8 companion articles in the same journal. They kept doing what I had shown you earlier, but on a bigger scale. So in this case, for a whole fly brain. And well, I mentioned that the fruit fly is small. I mentioned that the head of the fruit fly is smaller. And now from the head itself, that's the head of the fruit fly, the biggest part would be the compound eye, which is just shown in gray. And the compound eye here is not shown, it's just the stuff inside. All right, and this has now, you can segment this, and this was accomplished by a mixture of a massive human effort. So you saw that amongst this list of authors was the so-called Flywire Consortium, and they tried to gamify this and make this a citizen science experience. So there was this interface, and you could, or you still can, fly through these 3D stacks and say, yeah, now this process goes here and goes there. And they decided to use this data that I'd shown you earlier that was created by Davi Bock, the kind of data that most other people had given up on as being too hard. The data I had shown you from Davi was from mouse, but here they've done this for the fruit fly. Okay, and I have, okay, so this has been very recent, late 2024. And so we now have a connectome for the fly brain. Some of these articles now study what this connectome means, but it's a long way before we're able, before we'll be able to do that on humans. All right, I have another pub quiz question for you. So among the gentlemen in the audience, who thinks that you and I and Davi Bock and all the other gentlemen on this thing have the same grandfather? And who of the ladies in the room think that she and Ada Lovelace, who invented computing, and any of the female authors on this list, that you have the same grandmother? Who believes this to be true? Some of you. Okay. So the scientific consensus is that it is true. Yeah? And let me try and explain. It has to do with this. So here we see schematically the chromosomes of human beings. We have 2 copies of each chromosome. And then if we are female, we have XX, and if we're male, we have XY. And now when 2 people meet and they have children, then there is a sperm involved, and the sperm has one of these 2, and then one of those 2, and one of those 2. So from the father side, how many different sperms in sense of DNA are there? Well, it's 2 times 2 times 2 times 2 times 2, and so on. So 2 to the power of 22 or 23. And then from the mother's side, it's the same. The egg can hold, a copy of this one or of that one, or of this one, of that one, and so on. So this explains why we have quite a bit of diversity. And by the way, even so even identical twins are not perfectly identical in their genetic makeup, because there are still some variations in copy numbers and so on. Anyway, there's this. And so men have this, and this Y chromosome they can only get from their father, and the father could only get it from his father, and the father could only get it from his father, and so on. And that doesn't hold for the X, right? Because we can get an X from either father or mother. However, remember the mitochondria. Actually, the sperm has mitochondria, and the egg has mitochondria, but when they meet, the sperm's mitochondria are killed off and recycled, and it is In the humans, at least, it's only the maternal mitochondria that survive. So we all have the mitochondria from our mothers, and this is why there is indeed the notion of all the Ys going back to a single male and all the mitochondria going back to a single female. And this brings us to something called, because it's a machine learning lecture, to stochastic processes, notably Galton-Watson stochastic processes. And this is now not the boring, you know, throw a dice kind of thing, but it's a stochastic process defined over trees. And I've simplified it here because normally it's defined or it's studied in the rate of in the context of a Poisson process. So here we see, for example, the purple Poisson process has an expectation value of 1, and it means you know, there's a chance of having 0 or 1 or 2 or 3 or 4, or these numbers are not 0, by the way. It's just very unlikely down here. Now we could use this to model the probability of offspring surviving. Yeah? And if, for example, we use a mean probability of offspring surviving, that's 1.5, and you know, there is also the chance to have 10 or 11 or something offspring. To draw a nice diagram, I've here decided that each individual can have, you know, 3 chances at creating offspring. And let's now look at a process with a survival chance of 1.5. And it looks like so. And this could die out, but in all likelihood it won't. If, on the other hand, we use a survival chance of 1, then this is what Galton and Watson studied. This is guaranteed to die out. Yeah? So let's take this realization here. Looks pretty good so far. Yeah? There's 1, 2, 3, 4 descendants alive after 5 generations. But this is guaranteed to die out. So this Galton and Watson proved mathematically. Yeah? I'm not going to go over the proof here because it's more than a few lines. And so if we think of all the males alive at any one point in time, and at the offspring that they produce, let's look only at the male offspring, then eventually most of that offspring eventually will die out. And the same reasoning goes for the females. And hence this notion of all of us living humans going back to a single male, most recent common ancestor. It's also called the Y chromosomal Adam, and all going back to the same, and all women going back to the same female most recent common ancestor. That's the mitochondrial Eve. And they have never lived, they've probably never met. They probably lived a few thousand years apart in time. They definitely lived in Eastern Africa. And yeah, it's around 200,000 years ago. And yeah, I think I will, and this is, of course, I mentioned earlier that most of us have some Neanderthal in them and so on. So there was horizontal gene transfer. So we are genetically diverse. But I think I will leave you with this Christmassy thought on all of us being brothers and sisters, ultimately. And yeah, with that, I wish you a wonderful Christmas vacation and looking forward to see you next year.